Alright, good morning everybody. While we're setting the projector, so let me just go a step back and where previous speaker left you. I think in your PhD journey, I think most of you are probably aspirant of doing PhD or research scholars somewhere. I think you'll always find, it seems very exciting when you join PhD program, whether within India or anywhere when you go abroad. But what actually becomes after that, the first 2 years becomes really challenging, right? Anywhere you go, because immediately expectation is like, you were trained in the small pond and now you have been left in the big ocean independently, right? You have to now swim and survive. Expectation is that you know everything now and you have to be handling the project independently. So that is the time when I think your actual, not only your hard work, but also your ability to cope up the stress is going to really dictate your success, right? So I think the previous motivation lecture was very relevant for you to think about how to not only do good research and good hard work, but also keep up the good health, both physical and mental, and have the patience and perseverance. Those are the skills which I think will take you a really long way. PhD is not a small degree program which you just being measured with your few coursework and credit program. It's actually going to be taking you in a very long journey. And the first 2-3 years are very, very difficult because most of the time you start excitement, you suddenly see the excitement goes down. Then you will start thinking quitting programs, maybe think that okay, it was probably not meant for me. But again, think about your goal for which you plan to join the PhD, and the much bigger vision, much bigger picture. Within that, then the small problems will move away from your road, and you will succeed in that way. I think everybody goes through that that cycle. It's a good idea for you to really prepare yourself very well in this direction. So let me start with the small clip, which is one of the first big project happened in biology area, which I think in front of most of us, which was being announced in White House. So let's start with this video clip first.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. The moment we are here to witness was brought about through brilliant and painstaking work of scientists all over the world, including many men and women here today. It was not even 50 years ago that a young Englishman named Crick and a brash, even younger American named Watson first discovered the elegant structure of our genetic code. Dr. Watson, the way you announced your discovery in the journal Nature was one of the great understatements of all time. This structure has novel features which are of considerable biological interest. Thank you, sir. How far we have come since that day. In the intervening years, we have pooled the combined wisdom of biology, chemistry, physics, engineering, mathematics, and computer science. Tapped the great strengths and insights of the public and private sectors. More than 1,000 researchers across 6 nations have revealed nearly all 3 billion letters of our miraculous genetic code. I congratulate all of you on this stunning and humbling achievement.
Alright, so this was actually one of the first big announcements made at White House, when first time we thought that we can now understand all the secrets of human life, and first time all the human genes were sequenced, published in 2 different parallel projects with the both public and privately funded published in Nature and Science. And this was the first time which we also learned that we are not very different than chimpanzee. We are not very different, in fact, than many other model organisms, right? So, for example, C. elegans, yeast, Drosophila, we are all having the gene numbers, in the ballpark of around 20-25,000 genes. So immediately the entire focus shifted that how then we are so different than many of these organisms and what makes us different. So it means in the central dogma of life, some molecules are much more important, which gives us that ability and intelligence and different phenotype. And immediately after the genome sequencing projects, the focus shifted to the proteomics and field of understanding the big omics sciences. So with this, let me start my first lecture, and let me first thank Dr. Arup for inviting me here in this GAN program. I'm sure you're going to have a very stimulating experience in the next few days, but it will all depend on how interactive you make it. And it should not be just one way. It should, be very open discussion, because utilize this platform to really clear up your doubts and interact more with the speakers and your resource people here. So I thought, while I'm more specialized in the proteomics, metabolomics, and other areas of functional biology, but I thought to start with a little bit basics of genomics, because that is the foundation from which the entire big data sciences and the omics sciences actually started with. And then where we are currently heading with all this knowledge sphere of omics, moving toward personalized medicine. So I come from IIT Bombay, which I think some of you are aware of. It is one of the India's higher institute for research and excellence, especially institution of eminence status we have. You are welcome to come to some of the program and training which we conduct. I think I'll give some update towards the end. I come from Department of Biosciences and Bioengineering, where there's a lot of focus on the post-grad education and research. Almost 200 research scholars, 250 are doing PhD, and very strong program in the interdisciplinary research area. I have established this national facility for doing the proteomics research and training program at IIT Bombay, which is again funded by Government of India. You're welcome to come, and actually you're entitled to send your samples and analyze and make use of these resources, which we very much take from all external users. This facility is equipped with various type of Orbitraps, and we regularly conduct some training workshops which can be really useful for you. Given the pace of how the proteome should be analyzed in much faster ways one could do the sequencing, we have recently set up this new technology, which is the timsTOF based mass spec, for doing the 4-dimension separation and 4D omics at our startup, which are mentioned Proteomics International Private Limited, which was just inaugurated last week. And that will give us now much more ability of sequencing the entire proteome at very unprecedented rate, which was not possible earlier. Even we are very surprised, like how fast you can do these experiments. Typically, we have estimate of human genes around 20,000 human genes, what we have. And earlier it was thought about around 25 to 26,000 genes, but after pseudo genes and all were dropped, then finally 20,300 some genes we have. But from those, how many actual protein we have, there is no clear estimate. One would estimate that at least there should be 20,000 protein directly which is formed from the genes. But eventually with all type of alternative splicing forms and the post-translational modification, one would expect maybe 1 million forms of these functional proteins. How to really sequence these proteins at the fast pace and look at in totality. So that's where the complexity comes. Unlike the DNA with the 480 GC letters, now when we talk about 20 amino acids and side chain chemistry, it's not straightforward. Not one method, one technology can really do everything in a very, very rapid manner. So that's where I think there's a lot of advent of new technologies which are taking place. And I think in my first 2 lectures I will try to walk you through about where we started and where we are currently stands for, and where the more opportunities lies in the future. So my lab does a lot of work into different technology platform and assay development, different type of software and databases which we develop for the public, and also we have done a lot of work in the brain tumors and other cancers, and some work on the infectious disease area. But again, as I said, I'm going to make it more basic lecture, and aim is to give you gyan in not the research topic to tell what we have done, but rather the basics of the technologies and tell you what you can do from these. So genomics started with looking at the ambitious goal of sequencing all the genes. Again, that became very first effort of looking at a big biology project in a very systematic manner by 15 countries together, with thousands of scientists coming together with billions of dollars being invested. Goal was only simple, to look at the sequence of one human, right? But then after that, then we realized that we need to really study the gene expression, looking at the protein expression. That is going to dictate the biology, right? So that's just how I'm going to talk to you about some of the technologies in this space of genomics, moving to transcriptomics, proteomics, and multiomics. So genomics project was really remarkable, gave us the blueprint of life, but that in a way turned out to be slightly disappointing than what was estimated. We really thought that we were going to unravel all the secrets of human life after the genome sequencing project, and that was the time when I was actually planning to start my PhD journey in 2001-2002. But I was very excited when I was working already samples on the proteins, and I was very excited that now the proteins are in the forefront, because after genome, everybody realized that we had still a lot to learn, for the complex human system. So proteomics is going to be the next data science field, which is going to give us more information about the secrets of life. So that's where I think then I chose my career. But again, genomics also gave us not only the first time the genome sequencing ability, but also the high throughput sequencing technologies, also the way to work on the big data sciences, and first time developing the databases for the human, which actually served out the reference genome even for the protein structure. So that was not just doing the genome search, it actually really impacted many different ways of our understanding. And this Human Genome Project became one of the flagship projects of the entire human history, when the aim was to not only look at the human DNA or the sequences, but also look at the other model organisms, trying to sequence along with the human, and can we really identify all these tables of the genes and store them in a database form, make them publicly available because it is a publicly funded project. And also then we started realizing that big data biology will have its own legal, ethical issues and how to really handle that, because now we are revealing everybody's secret of life uniquely, and are we going to make it public? So if you sequence the genome, how are you going to handle that? And are you going to really reveal the personal disease, the racial information, all kind of, because people have a lot of sentiments around that. So again, that also brought in many new aspects of understanding that biology is not alone. You have to think about many social aspects as well. So this first genome sequence project was actually done on the 20 individuals. One of them was from the blended ancestry, which was essentially American with some Asian. But this was again in any biology research, as you know, we keep the anonymous stuff, right? So we don't reveal what the patient ID is. So this was again anonymous, which individual were sequenced, and 19 were from the European ancestry. Together with that entire sequences, it was put together a reference genome map, which is what being used to search against all the human genes and all the human proteins. Idea was simple. Can we take every chromosome, cut those into the small pieces, clone each fragment, amplify them, and sequence those small fragments and put it together in a logical way so that now we can start getting the long reads. But it was the very beginning of the entire genome sequencing efforts, which again, if you look at timeline of biology project, it goes decades, right? So that's where you need a lot of patience to really see good fruit of your work. This project started in 1984, and 1989, Dr. James Watson, he became director for that project. Eventually, 1990, they started a 5-year plan for human genome sequencing. 1893, Francis Collins became director. 1994, they published first genetic linkage map. Eventually, then they came with the first pilot project of human sequencing. In 2000, you saw that it was actually announced that now we got the first draft human genome map, not the complete sequencing, but the draft genome map, which actually got published in 2001. And 2003, it was announced to have it completed. One scientist which made huge contribution to this was Craig Venter, which along with the publicly funded project, they did privately funded. And this was really very fast track, which they finished in a very almost similar time than the public funded, and both published in same year Nature and Science. So why this project was very a milestone, because not only it gave us just the sequences of human gene, but also in many different ways of our ability to think about our environment, our health, and many other aspects which is now dictating, and now we can realize that the long-term impact of those has reached to the precision medicine, looking at different type of the treatment therapy, right? Everything is being dictated with those just knowing at the sequence of these model organisms. And immediately, as soon as we got the genome sequences, now landmark studies started coming in. For example, we had prediction about human evolution, but there was no credential evidences made. But now, in 2010, they sequenced the Neanderthal genome and found that, it was probably the closest ancestor. So now there's a credential evidence made. In 2009, again, there was a big study made on the brain tumors, glioblastoma. They sequenced all these patients suffering from the glioma and glioblastoma, and first time identified a very commonly known protein, isosaccharic dehydrogenase, IDH, which has a lot of mutation. And that became a landmark, now one of the key targets for classifying these tumors. So again, our ability of looking at different aspects of biology, from evolution to medicine, has totally, truly transformed with just this basic discovery. So how the sequencing was performed in a very crude way, basic way, the Sanger DNA sequencing was they started when they were looking at very, very short stretch of DNA sequences and then trying to put together, which helped us to first do the very small genome sequencing of the bacteria, Haemophilus influenza, 1995, moved with very other model organism from the east, Drosophila, Arabidopsis, human, and eventually mouse was even after human, got completed. Then this concept of Sanger was very difficult, so then they took, can we bypass the chain termination method and use now the next generation sequencing. And this is one of the technology, one of the area genomic sequencing, where you see last 2 decades, we have really proved all the predictions wrong, right? The pace at which now the genome can be sequenced, it took 15 years time for first genome sequence project to complete. Now it may take, maybe less than few hours for one genome sequence to complete. It took $1 billion to do that project. Now you go to any institute and in just, maybe less than 1 lakh rupee, you can do the entire genome sequencing. So think about the pace and the precision and accuracy. This is where the engineering discipline, interdisciplinarity has really contributed a lot, right? And we can see that how this technology has really surpassed the Moore's law and all the predictions. So again, looking at the next generation sequencing, the idea was, can we make utilize our different physical principle, how these ATGC bases, when they are going to release, release the inorganic phosphate molecule, and based on that, light can be now sequenced when the ATP, TTP, GTP or CTP, when they are binding, the flash of light is being utilized to sequence. But again, idea was this was a very iterative process. Can we now do much more high throughput manner? And then now the third generation sequencing technologies came. And therefore now idea was, can we utilize the entire stretch of DNA for sequencing? Can we take double-stranded DNA, denature it, make single stranded, let it pass through some ways for doing sequencing, and now a lot of physicists, engineers came in forward and started utilizing what is the difference between ATGC. Can we look at their physical property and try to map these base pairs? And now one of the technologies is nanopore sequencing. It just looks like in the membrane porous space, how the ATGC, when they pass, they have different perturbation. They do different type of electrical conductance change, and based on that, can you now measure ATGC in a very, very rapid manner, which is now being utilized with a very, very handheld device, a small device, which can be sent to the rural areas. Any place you go and take this small like hard disk type of, the nanopore sequencing device, and then do the genome sequencing, send the data back to server, and now it can be, it can do the genome analysis anywhere. This is just one of the technology. Of course, I'm not advocate of any technology, but giving you like how technologies are getting more miniaturized and robust. But much more, specialization is coming from Illumina technology, which is one of the leaders in this area, and so as the Thermo Fisher, where they have iron current technology. We are not genomics lab, but often you have to correlate the proteomics changes with the genomic mutation and lot of alteration at the genome level as well. So we also have one of the iron torrent genome sequencing technology. In this one, I think one of my PhD students will show one of the setup we have to sequence
The instrument behind me is the iron torrent S5. This instrument detects the proton ions released during the formation of phosphodiester bond as a signal for sequencing. It comes with 2 other smaller instruments, which are iron 1touch2 and ion enrichment system. The first step in a sequencing reaction is library preparation. Once that is done, we use the iron 1touch2 system for clonal amplification of the library. After the library amplification, enrichment is performed on the ion enrichment system. It uses biotin and streptavidin interaction to select the template positive ISPs. The last step is to start the sequencing on the iron 1touch S5 instrument. For that, we load the sample onto the chip. The sample should be uniformly loaded on the chip, and then we place the chip on the chip clamp. We close the chip clamp and then we check that the washing solution, cleaning solution and reagent cartridges are in place. After that, we start the sequencing.
Okay, this is one of the very simple way of looking at genome sequencing for millions of base pairs and with the massive sequencing one could achieve, but we do more for the targeted work in this area. Another technology which is very much gaining momentum and news is the CRISPR technology. You might have heard the last year, 2023, the first CRISPR technology for the sickle cell anemia got approved, right? And again, like, our ability of editing genome, sequencing genome, modulating genes is now started truly revolutionizing our ways of treating the diseases. Sickle cell anemia, like, looks very simple, but individuals who have the defect, their oxygen binding capacity is severely affected. And while the basic science research has identified there is some important gene in the BCL11 which is going to be very important to as a target which can be utilized, but our ability was not there to deliver it into the very, very right form of editing. So CRISPR, it came again with the basic knowledge, looking at the cluster of regularly interspaced short palindrome repeats, which is identified in bacteria. An idea was from this bacteria uses CRISPR for its own immune system, its own defense against the bacteriophage. When different viruses infect the bacteria tries to build its memory by adding some of these spaced sequences. So it's already had the repeat sequence fragments, but it adds, let's say, one bacteriophage came attack, it actually adds a spacer from the degradation product of that phage DNA. It added that DNA sequence in its own genome to add the memory of it. Likewise, another phage infected the bacteria, and bacteria cast protein, which is actually now killing the phage DNA. It's actually after that it brings the DNA into the bacterial genome and now makes it another part of the memory. So now next time when the phage infect bacteria is better prepared to kill the phage. So it has actually utilized this particular system for its own benefit and advantage. But now scientists thought, okay, can we utilize the bacterial learning for the CRISPR-Cas system and utilize for the treatment? And that's something which got Nobel Prize to Dr. Emmanuel and Donda for doing the CRISPR-Cas technology in 2020 Nobel Prize. And that immediately started showing the big impact. Now we know, like in the human growth, when we have this protein for the sickle cell anemia, which is having the mutation, that happens like if you're a little bit familiar with the hemoglobin protein and the myoglobin protein, right? So in hemoglobin there are tetramer, alpha 2, beta 2. So one of the beta protein gene that gets just one base pair change, and because of that the protein totally becomes like sickle cell shaped. So in our childhood, a protein is for the hemoglobin known as HbF, hemoglobin fetal, and adult known as HbA, which is hemoglobin adult. Now that transformation from the fetal to adult is transformed with the transcription factor BCL11A. So now what people thought, can we utilize the BCL11A gene, because that is very efficient, because in the childhood, inside the womb, the embryo has to totally do the very efficient oxygen binding inside the mother. So that HbF is much higher binding capacity than HbA. So then people thought, can we now utilize this BCL gene and try to change the course of when at the time of birth HbF level goes down and HbA level goes up. Can we find out this BCL11 switch and try to control that, modulate that, so that now HbF level remains higher in these individuals, and that has led to the cure of sickle cell anemia, which is, I think, amazing. And this is just the beginning of many of the remarkable discoveries which you are going to see in front of us, when a lot of clinical already trials are going on for different diseases, and something which you will be witnessing more and more, how these understanding and editing technology are going to impact. There are many ongoing efforts in the genome. The first one which I showed you was about the 20 human sequences which were made as a reference genome. But think about it, did not have the Asian representation. So whatever you were searching in the genome, it was more about the Caucasian data. But now then they added 47 people and more recently 350 people. So again the genome references are getting enriched with variety of population. And more so, it is now every country started realizing the power of the genomics revolution and started launching their own program. And India is not much behind in that. Even we have the 10,000 individual genome sequence which already got completed in March, and more of the secrets are going to be out soon. Additionally, we have a national cancer bridge connected from Tata Memorial Hospital in Mumbai, which now connects all the different states and their nodal centers, and trying to collect all the digital information about the cancer patients, trying to utilize that information pool at one place, which is again, it was not possible previous years. All the clinical research relies on clinical data. And such type of initiatives are very, very important. Kerala, being very progressive state, started actually in fact sequencing the population which are more prone for sickle cell or malaria or dengue. And now they are doing these endemic populations in the Wayand and Attapadi, these provinces. They are looking at the genome of these individuals which are more prone. What makes them more prone for the getting infected with the dengue or sickle cell? And again, these are just population-based screening at the genome-wide level, which is going to really transform the healthcare system. So again, many of these are just happening in front of us, and I think you're all part of it. But I thought to emphasize you where we started a big data sciences, the genomics, where we are leading with the new and new developments in the field, from the Sanger method to the NGS method and many type of new sequencing technology, from nanopore, Illumina to the ion torrent, etc. How now this information, which is the base reference map, is going to lead us towards understanding much more functional content. So again, the looking at from the DNA to the RNA form, and again you are all familiar with the reverse transcription PCR or real-time PCR and impact of that during the COVID time, when we only knew some of the sequences of the virus. From the patients, the mRNA was extracted, cDNA converted, and now that is sequenced for the specific genes of the SARS-CoV-2, but same could be utilized for many other applications. Likewise, you can always look at where the gene is being expressed in a technique known as in situ hybridization, which is essentially very elegant way of utilizing microscopy and our understanding of different gene expression and making those probes, and then trying to see where the genes are really expressed. So in Drosophila model, they have shown to look at the developmental stages. If you have the probes for looking at different genes for the wingless and engraved genes, which is involved in the abdomen and the wing, where they are being expressed throughout, you can see very beautifully how the mRNA is being expressed. And for more and more gene, you have more probes, you can see live where the gene is being expressed. So again, the C. elegans and zebrafish and these Drosophila are very nice model when you can visualize the gene expression in the live cells. Right? Gene expression in the high throughput manner is another thing which is being thought of. Can you look at entire genome-wide, proteome-wide expression of these genes, and the small chips on which we have thousands to millions of features printed, can utilize them for looking at the gene expression profile. So idea is, if we have this gene microarray chip, if let's say you are comparing the cancer cell with the normal cell, you extract the RNA from the cell, tumor cell and the normal cell, you convert them to the cDNA, and in the process you label them with different type of fluorescent fluorophore, Cynin 5 or Cy 3, and then you mix them together, and based on the principle of hybridization, if the DNA was printed on the chip and the cDNA is going to get bind to there, you are now going to see the expression of these genes. And what you see at the end, you are seeing some of the very colorful dots of green, blue, red, and the the gray. And what it tells, because you have labeled with Cy 5, Cy 3, this is probably highly expressed in the diseased tissue. The red ones are showing, the green ones are showing you it's much more in the normal, it is down regulation in the actual disease condition. Or both of them are showing the same pattern, it means it is going to be no specific expression for the disease, or the gene was not expressed. So again, just by looking at this profile, why it is so important? Because now first time we learned that breast cancer is not one cancer. Just you look at just the gene expression profile, women who are having different type of hormonal receptors and these kind of unique gene expression, they make them 4 subtypes of luminal A, B, HER2 and basal live. And based on that, now we know that with estrogen receptor or the progesterone receptors, herceptin receptors, or all of them being absent in the triple negative breast cancer, that is very unique. And if you are giving the same chemotherapy to every woman suffering from this cancer, it's going to not respond same way. And then that's why we see so much deleterious side effects, right? So now if individuals can be sequenced beforehand, they will have the probability of understanding the disease ahead of time. Doctors can treat them much more personalized way. And again, these sequencing technologies can be also truly revolutionary, and sometimes celebrities bring a little bit more knowledge to the public in a way which you will see in this clip.
Her face is known around the world, and now her secret is known too. Angelina Jolie revealed in a New York Times editorial Tuesday that she had a preventive double mastectomy earlier this year, after discovering she has a gene defect that greatly increases her chances of getting breast and ovarian cancer. She writes, "My doctors estimated that I had an 87% risk of breast cancer and a 50% risk of ovarian cancer, although the risk is different in the case of each woman." While Jolie's genetic risk was very high, cancer experts say it's also fairly rare.
So 5% of breast cancers in the United States occur in women who have the genetic mutation, the BRCA1 or the BRCA2 gene.
Doctors say women with a family history of breast or ovarian cancer at an early age are more likely to carry the mutation, and insurance companies usually pay for genetic testing. For those who test positive, Dr. Rachel Brem says having both breasts removed like Jolie did is a very personal and difficult decision. It should be made with the help of genetics and cancer specialists.
It doesn't mean that that's the right decision for everyone. And the important thing to do is to have frank discussions.
Jolie said she went public because she wanted women in her shoes to know they have options and can still look and feel like a woman after a mastectomy.
Well, I think one of the reasons that we're seeing an increase in what we call bilateral or mastectomy on both sides is that there are better reconstruction techniques.
The actress was treated in a California breast cancer center.
We hope that the awareness she is raising around the world will save countless lives.
Jolie's star power is already drawing attention to a complicated choice some women must face. Kelly Dashel, Associated Press.
Alright, so again, looking at so her family history was her aunt and mother to perform those functions. So the body conceives those signals to where to do what exact activity to do, right? The genes are there, but they are going to do specific transcription translation towards that activity. Therefore, measuring this dynamic perturbation of transcript and protein become much more crucial than just the static blueprint itself. And looking at the spatial, the cell type protein information, or looking at the temporal different time change protein, even circadian rhythm people are studying, all that is really crucial to really understand the physiology of a given system, right? So proteomics, what do we study in the proteomics? Now question to you, what all do you want to study in the proteomics? And what do you achieve, what do you aim to achieve from proteomics? Please raise some hands. As I said, it should not be one way, right? Because now it is a small group, more interactive.
I think those who are sitting at the back can come back.
No, everybody, we should give opportunity. I'm sure you know that. I've already given you a lot of context here. Based on this, why do we study proteome? So Dr. Arup is going to, he's trained, well trained at IIT Bombay. Now he's going to conduct some quizzes as well to understand your knowledge, how much you are able to absorb, right? And as I said, please make it more interactive, because that's the only way you will take something back, right? It should not be one way around. My lot of lectures already on YouTube and Gyan and many other portals, including this IM portal.
Sorry to interject. So we are having Professor Sanjeeva only for this session. He has lots of lectures filed up at IIT Bombay as well. So I think you better use this opportunity to interact with him. YouTube lectures are there, but you cannot talk with Professor Sanjeeva over a YouTube lecture on YouTube. Maybe we are a couple of decades back, maybe in 10 years time you will have that facility. But right now I think if you're raising your hand, yeah, please.
To understand the structure of protein and how proteins are organizing, what are the dynamics they are creating, and what are their folding statistics. So that is why I think
proteomics, we can also like study
Into the hospital and started on very strong chemotherapy. Unfortunately, though, the leukemia didn't go into remission after being treated with this very high-dose chemotherapy. There were no real promising treatments on the horizon, and so things looked rather bleak. But at the same time, as I was being treated, my leukemia genome was being sequenced by the Genome Institute here at Washington University School of Medicine. We got back the sequencing results, which showed that there were promising targets that weren't even in the scope of our standard treatment protocols for this type of leukemia. Because of those results, I was able to start less aggressive chemotherapy that I could tolerate. and then targeted therapy that had been approved for actually renal cell kidney cancer. Within a few days, my blood counts improved, and within a few weeks, my leukemia was in remission. So it was really going from a situation that looked really bleak to now a situation where there was hope again.
Alright, this is one of the great example I see because I followed his study in 2012 when he formed and doing research. New gene defects which can be controlled with different drugs very effectively if we know our precise information. And again, as I said, these kind of transformations started happening in many places, especially in US. Like in one of the Michigan Center for Translational Pathology, the way they do the patient, like on the day one, for example, let's say tumor biopsies are being done. Pathologists will take the sample for looking at the routine pathology images, and then the samples are being prepared for the RNA, DNA, protein, and then different type of sequencing methods are being used to analyze the data. Then the sequencing tumor board consists of these doctors, clinicians, and the omics data scientists. Everybody together looks at one individual in its uniqueness and look at what all structural rearrangements happen, different mutation happened, gene expression happened, and what should be the best treatment given to that individual. So now, in 1 month is almost required, 4 weeks after doing the surgical resection to recover anyways, and during that time we are providing the patient a very unique way of treatment. This is really transformative, and lot of work and lot of good publications are in that area. Again, like the way you are going to be more exposed about artificial intelligence, machine learning approaches is going to really help us to narrow down the choices for clinician. It can't replace clinician, but it's going to really make them better informed and give them better options to choose. Look at the different genetic disorders, even cancer subtype detection, predicting disease progression much ahead of time, and looking at analyzing genomic variants, etc. I'll give one more talk, but I may not have sufficient time to give you a lot of my research talks to give today as well. But just came to mind that during the COVID time, Arup was in our lab and Professor Graham was working from UK, and we were working collaboratively. One problem which we were looking at, that how different patients are, everybody is mild form, but some people are getting to severe form, right? So we were, the clinicians were very much in a difficult situation to whom to prioritize and give them the bed and do a ICU or the hospitalization, because everybody is panicked and those who are more influential will get the bed. But rather than that, can we look at the markers, like who really needs the support, right? So they were looking at some indirect markers of looking at the various type of biochemical features and looking at their IL-6 level, interleukin-6 level. Many of these things were giving them indirect information, but no credible tests were being made. So we were working on the COVID-19 research, and Dr. Rup was part of the team who was looking at the entire blood plasma proteome profile of these individuals. And the team identified the big signature of the protein which is changing from the mild to the severe form. But now which among those could be the actual target to really develop the test? And that's when Professor Graham came in touch, and he did machine learning to say where the major nodes are coming from these the data cloud which we generated, right? And based on that we identified 4 major proteins. Apolipo-B was one of those, and we have the fibrinogen gamma chain protein and serpin proteins. So based on these proteins together, we were able to now predict individual much ahead of time that how one could actually progress from the mild to the severe form. And that was the first test which got patented, which is uniquely able to make prediction about a COVID patient at the mild stage, maybe moving towards the progressive stage of the severe child. So again, the artificial intelligence machine learning models are going to give you the prediction ability much more accurately based on your data models. It cannot just happen just out of the blue, but it has to be based on the credential data sets. Again, one look for more non-invasive methods for cancer detection. Can we look from the breathing? Can we look from saliva? Can we look from our sweating? Variety of ways. One is thinking urine as of course another sample type. Blood is still invasive, but could be used for detection. Many of these sample types can we really do much more accurate ways of understanding disease and even looking at better cures, right? So I hope my first lecture gave you the at least set up a stage for your entire Gyan program, how these various omics technologies have really, which is happening in front of us around society, around your clinics, has really started giving us better ability of looking at every individual in much more detailed, in-depth manner, and everybody can get better, kind of disease prediction or better treatment by utilizing these technologies. And over the time, with more and more good models being created with artificial intelligence, probably our prediction ability will also keep improving, right? But you need a lot of actual data set to work in that area. With this, let me thank you for being very patient and having at least some good questions. I'll stop here. If you have any questions, please ask me. Questions, comments, anything which you want to share?
Good afternoon, sir.
Hello.
Sir, you told us about L01 gene, which has helped us for treating sickle cell anemia as it helps in hemoglobin switching. So, sir, may you please throw some light upon this technique of how this gene was used or manipulated in order to treat those patients?
That's right. So, very good. There are two different things that are happening here. One is, of course, the basic science research, which discovered the role of important proteins and important genes, right? Which people have done differently. Like, they were looking at hemoglobin expression and what makes the fetal different than the adult form. That actually led to the discovering of this particular transcription factor, which was expressed at the switch off from moving from the fetal to the adult form, right? But then we got the CRISPR-Cas technology, which gives us the better ability of doing the gene correction, gene editing. And since we know that in the beta gene of our complex of alpha 2 beta 2, that defect can lead us to this problem of the sickle cell anemia. So idea was, can we utilize this particular gene which makes a protein for very efficient way of oxygen binding in the embryonic stage of the fetal, and use that gene to correct the defect for the adult, right? Because this gene which makes the protein for the HbF is going to give very strong oxygen binding. So the two things were brought together with the treatment of this technology, but were discovered separately. So like that, the CRISPR is just an editing tool which is going to utilize many knowledge of different basic science labs for different diseases, but now how to deliver them very accurately and very precise manner, do corrections. Any more questions? Alright, Arup is not here, but I think we thought we'll give you 5 minutes break just in case you want